before we can all start the whole process of mitosis, we need to start with the interphase. Interphase is something that happens 90% of the time of the cell. So what happens there, the cell grows, it duplicates DNA, and also it forms organelles that it needs for the mitosis. So all of these is for the preparation for mitosis. And when it has done all this, it can start doing mitosis. So it starts with a prophase. In the prophase, the spindles form at each side. The nuclear envelope starts to dissolve, and you can see the chromosomes better because they're more condensed than before. Then after that you have metaphase. Metaphase is where the nuclear envelope is completely dissolved, and all of the chromosomes have lined up in the middle, and then the spindles kind of go out and grab onto the chromosomes from each side. And then in the anaphase, the sister chromatids, they go apart, sister chromosomes. So we have, and they're getting pulled to each pole. So like this. So they're getting pulled in each of their own directions. And then in the telophase, it has then got them pulled on each side, and they form each their own nuclear envelope around those chromosomes. So you will have kind of two nucleuses at this point, but then in the end you still have the cytokinesis, which is where it kind of splits up into two daughter cells with both the same DNA and the same, but completely different. And you use mitosis for example, if you grow, then you need more skin cells and such stuff, and for example, to repair something. So if I have gotten a paper cut, then the skin kind of needs to repair and close. That was mitosis.
Okay, so I'm going to be talking about meiosis 1. So meiosis 1, it has 5 different phases. The first phase is prophase 1. So here is the cell membrane, and in here is the nuclear membrane. Here you can see the centrioles here and the centrosomes, and they're around here. Here are the chromosomes. Here's the chromosomes, you can see here, and the sister lipids here. The sister lipids are here. Here what happens is the crossing over happens, and the chromosomes, they pair up. For the next phase, it is metaphase 1. In this phase, the nuclear membrane, it breaks down, and the centrioles, they move over to their poles, so they move over here. And the DNA, the lipids, they attach to the chromosomes, and they slowly start to pull onto the chromosomes, pulling them over to their end of the poles. Here for anaphase 1, what happens here is that the centrioles stay in place here, and the chromosomes get pulled here, still attached. The chromosomes get pulled, and basically the chromosomes, they get separated. As you can see here, they get separated. And here we have the next one is telophase 1 here. Here, this is where the actual cell starts trying to split apart, and what's called the nuclear membrane, it reappears. And as you can see here, it reappears, and it goes back to here, and here are the chromosomes, and the sister lipids are still here. And the last phase is cytokinesis. This here is where they finally split apart. And here it looks like this: the nuclear membrane, the cell membrane, and just the actual cells, sister lipids, everything here. This is how it looks like. This is how it looks like. So basically, the sister lipids, they are used for copying the DNA from the original cell, so that the new cells that when it gets divided, they can have the copy of the DNA. So yeah, so it looks like that. And also some similarities for mitosis and meiosis is that the nuclear membrane breaks down so that the chromosomes, they can move around. And yeah, that's it.
Meiosis second. After completing meiosis 1, we go into interphase second, that's a rest period, and then we go into meiosis second. The first step of meiosis second is prophase second. Here your nuclear envelope dissolves again, and now we will go to the second step of meiosis second, that is metaphase second. Here, the centrosomes have now moved to the poles of the cells. Its nuclear membrane is now disappeared. It has the chromosomes going to the equator side. The centrosomes are actually connecting to the chromosomes. Go to the third step of meiosis second, that is anaphase second. In this phase, it begins the sister chromatids that are going to split, so they will become two daughter chromosomes after that. Now the separated chromatids are pulled towards the opposite poles of these cells by the spindle phase. It ensures that each new daughter cell will receive. Last step of meiosis second, the telophase second. In this, the chromosomes reach the opposite ends of the cell. Nuclear membrane reforms around each set of chromosomes. The chromosomes begin to deform, turning back into a diffuse chromatin state. Cytokinase follows and starts to divide the cytoplasm and results in 4 haploid daughter cells. These 4 stages are really crucial for ensuring that each gamete has the correct set of genetic information. The difference between mitosis and meiosis: the purpose of mitosis is to grow during development or to repair or damage, whereas the purpose of meiosis is to create opportunity for genetic diversity in the daughter cell. Mitosis has 2 diploid cells with identical genetic information, whereas meiosis has 4 haploid cells with different genetic information. In the end result of mitosis, it creates 2 daughter cells, and in the end result of meiosis, it creates 4 daughter cells. Mitosis has 1 cell division, whereas meiosis has 2 cell division. Mitosis has 46 chromosomes and meiosis has 23 chromosomes.